What I'm going to primarily talk about today is what I do in the Pulse crop pathology program to support a lot of the breeding activities that occur both in Lethbridge and in Canada. So what we're looking at here is what we call our Pea Disease Nursery. And we have lines here from really the 4 breeding programs in Canada, or that develop lines for Canada. So we have lines from AFC Lacombe, we have lines from CDC Saskatchewan, and then we have lines from a private company in Edmonton, DL Seeds, and then there's also a company in Netherlands that sends us their lines for evaluating different diseases. And we assess all of our diseases in just this one nursery. And really when we think of nurseries, it's kind of a learning tool on everything not to do, because we're trying to get as many diseases as possible as we can in this nursery. And so the 3 diseases that we're really looking for here is Fusarium Root Rot, Mycosphorella Blight, and powdery mildew. So how many of you here grow peas? This should be the first question, yeah. And how many of you know that the peas that you grow are always going to be resistant to powdery mildew? So that's really the one success story that we have with peas, is that we have good resistance to powdery mildew. So every line that is entered into this disease nursery should already have resistance to powdery mildew, and we're just double checking that it is indeed correct. So what we do is we plant a susceptible powdery mildew line, kind of randomized throughout this plot area, and then we just check to see that that powdery mildew line, yes, is getting powdery mildew, and all the other lines that are in this disease nursery are not getting powdery mildew. It turns out powdery mildew doesn't really like to grow in southern Alberta, so we have to really coax it along. So we give a lot of irrigation to these plants, and we also grow powdery mildew susceptible lines in the greenhouse. They get full of powdery mildew, then we bring them out and inoculate those powdery mildew susceptible lines to make sure that we can accurately assess whether there is powdery mildew resistance in these plants or not. So that's one's actually a pretty easy one to deal with, and it's a pretty easy check. Unfortunately, way too early in the season to see powdery mildew in these lines, so not much to really see. And in fact, right now these pea plants all look really healthy. And then the other disease that we screen for is Mycosphorella Blight, and I'm sure all of you that grow peas are very familiar with Mycosphorella Blight. It's basically going to show up every year on your pea plants. You're probably spraying for it, usually around canopy closure or early flowering is kind of the standard practice. There isn't a lot of resistance to powdery mildew, but we screen for it anyway. And again, we're trying to encourage Mycosphorella Blight. We're trying to encourage Mycosphorella Blight to develop in these crops. So we'll start irrigating once the peas are a little bit taller and the canopy starts to close. We're irrigating every other day on this location to really try and encourage that Mycosphorella Blight to develop. And like I said, unfortunately, we don't have a lot of resistance to Mycosphorella Blight in our peas. So it's really just seeing if there's any standout varieties that happen to have any improved resistance. But for the most part, they tend to be all equal to our susceptible checks. And then lastly, the other thing that we screen for here is Fusarium Root Rot. What we do to encourage Fusarium Root Rot, and I guess the same with Mycosphorella Blight, is that we actually are just planting peas on peas upon peas every year. So again, probably the worst practice that you could do as a producer, but it's the best tool that we have as a plant pathologist, is no crop diversity whatsoever. So we kind of have 2 locations. We kind of just jump back and forth, peas one year, peas the next year, and we just go back and forth, always planting on that pea residue. And that's really what is encouraging the Mycosphorella Blight, and that's also really what encourages the Fusarium Root Rot. So when we first started the nursery, we put Fusarium inoculum down the first year, and then just with successive planting of peas, we don't need to inoculate it for Fusarium anymore. It's just there in the soil. And we dug up some roots, and we'll kind of move over to the tailgate area once I finish kind of talking about here, and I'll show you what some of the early symptoms look like. It is a bit early yet to start seeing Fusarium root rot. It tends to come on later in the season, kind of at that end of the vegetative growth into the start of flowering. And again, we irrigate these plants a lot to make sure that there's enough moisture in the soil for the root rot to take effect. But we can start to see that it is starting now, and we have some plants there where you can start to see some of the little tiny lesions showing up on the roots. And so it's, you know, maybe still a good time to start scouting now, just to see if it's going to come on later in the season. And yeah, so that's basically the disease nursery. I was hoping when they said you want to do a plot hop talk, I thought sure, in June last year we had tons of bacterial blights in our disease nursery, so I thought, oh, this might be a good time to talk about bacterial blight. This year there's no bacterial blight at all. I think we planted a little bit later, and we saw a lot of bacterial blights in peas last year because 2016 was such a wet fall, if people remember that. It was hard to harvest, it was always rain, so I think a lot of the seeds became contaminated with bacteria. It got planted in 2017, it came out really early with the seed, but 2017 was such a dry year, we didn't really see any carryover of that bacterial blight. And it's usually not a big issue in peas. It just kind of popped up last year. I thought, oh, it's would be good to show, but we're not seeing it this year. So far, everything is actually looking pretty good for some of these early diseases from what I've seen in the pea crops. So that's kind of the spiel on the disease nurseries, and then we'll move over to the tailgate and I'll show you some real disease, since there's not much disease to see here in the nursery. Unless anyone has any questions first about kind of what we're doing in the nursery.
So you're looking to see any of these new lines have resistance?
Yeah.
And can you tell in just 1 year, or you have to look at them for 3 years?
They always are entered into the disease nursery for 2 years.
2 years.
Sometimes 3 years. If there's a line that looks promising and that maybe is like, oh, it's a new source of resistance, we usually check it again for a 3rd year just to make sure that we are confident in that rating. Or say like last year, 2017 was so dry, we didn't get a lot of disease even in our susceptible checks, then we might run it again for a 3rd year just to make sure that we are confident in those ratings. And I should say the nursery is also replicated at a location in Saskatoon, where sometimes they're a little bit wetter up there, so sometimes they have a little bit better conditions for disease developing. So when we're looking at this disease data, we're always looking at it from at least 2 locations and then also at least 2 years of data.
So are you seeing new lines that are sort of hopeful towards these 3 diseases?
Definitely with powdery mildew resistance, everything has. All the new lines have powdery mildew resistance. Mycopsorella blight, we haven't really seen much improvement in any of the new lines. They're usually, the disease levels are usually equal to our standard checks, our standard agronomy checks. Fusarium Root Rot, there is a couple lines that we have seen some improvement. What's interesting is those lines weren't specifically bred to have improved Fusarium Root Rot. It just seems to it was a surprise when we looked at it and said, oh, these lines actually look like they're standing up better. So those ones, I guess it's a decision of the breeder whether they're carrying those forward to be registered and if they'll make the claim that they have improved Fusarium Root Rot resistance.
Sure. Yeah. Is the Fusarium specific to peas or also hosted by the crops?
That's a really good question. Yes, so when we look at the Fusarium species, we are looking at a number of different species that cause the disease. The primary one that we're actually looking at 2 species that are inoculated in this field, Fusarium avinaceum and Fusarium solani, so kind of big Latin names. Fusarium solani is specific to peas. Fusarium avinaceum has a very broad host range. We find it on a lot of the other pulse crops, so it seems to really like lentils, it really likes chickpeas. Faba beans are kind of up and down on their reaction. And it can cause mild disease symptoms on cereals and canola, but not very serious disease symptoms. They might cause a little bit of root rot, but it's not a full-blown problem in those other crops. So it's Fusarium avinaceum is a pathogen with a very broad host range, but it seems to love the peas and the lentils, and the chickpeas is kind of its favorite food source. So it's not really of a concern in wheat and canola. You can just find it there sometimes.
Does that mean it persists in a rotation?
Yes, yeah. And that's something that I guess is just starting to emerge, this Fusarium avinaceum. Maybe we'd start looking at it as an emerging pathogen. Because if you look at literature from 10, 15 years ago, there's not a lot on Fusarium avineseum. And now all of a sudden we're seeing it pop up a lot more. And so I think we'll start to see that research is really going to start proliferating on that organism. And it's a pathogen that does survive on crop debris. A lot of pathogens produce overwintering resting spores. Avineseum doesn't actually produce any of these overwintering resting spores, so it really can only survive on crop debris. And it's been thought of more as kind of a saprophyte, as something that is just growing on crop debris, but now it seems like it's maybe switched over to a more pathogenic lifestyle. So there's a lot of questions around that organism. Its ecology, why has its kind of niche changed? Why does it love pulse crops so much? But yeah. Okay. Then we can move over to the tailgate party, and we'll talk about a couple of other of my favorite diseases. Okay. So first, these are plants that we just kind of dug up from the disease nursery, and trying to look for some evidence of Fusarium starting. And I can pass these around. There's not a lot of disease. They're actually looking very healthy. But on some of the plants, what you'll start to see, and this is how Fusarium root rot starts, is you'll start to see little tiny kind of brown, reddish brown lesions starting kind of around the point of seed attachment on this nice fleshy part of the taproot or the epicaudal. And eventually, it seems to take a long time for the Fusarium to kind of colonize that root and spread. But when we check these towards the end of June, early July, we'll see that has moved into kind of looks like a blackened kind of taproot. And so most of these look really healthy, but if you look closely, you can start to see some little lesions on the white part of the root. So they're small. I'll pass them around and see if you guys can spot them. There also seems to be some of them it's more maybe some sort of insect feeding or something on there. I'm not particularly sure what insect it would be, but look closer to the point of seed attachment and look for kind of really small red lesions. And this one is actually a really good example. I don't know if one little pea root can make it all the way around, but I'll see if I can find a couple more in here. Oh, I'm dropping some. Yeah, that was, oh. Yeah. There's another example of that kind of starting on the, and pass them over here. You just start to see, that one is a little bit damaged too, but you can just start to see some little brown spots on the roots, and that's really how it starts. This is Fusarium root rot, yeah. And my other favorite disease that we don't actually have a disease nursery for yet is Aphanomyces root rot. And I'm sure if people have heard me talk before or have gone to any sort of agronomy updates, things like that, I'm sure you've always heard Aphanomyces root rot. We don't have a disease nursery for it yet because some of the resistant germplasm is just starting to be incorporated into breeding programs where they're trying to move some of that resistance into agronomically advanced lines. So we don't really have anything to evaluate yet out in the field. The other issue is that it turns out that it's really hard to actually get Aphanomyces to establish in our disease nurseries. So we can go out into commercial pea fields. We've done extensive surveys. We found Aphanomyces is basically everywhere that peas are grown. We've tried to establish it in a field site here in Lethbridge, and we just cannot get that disease to take. So it's usually our story of plant pathologists. We get disease when we don't want it, and when we want it, we can't get it to happen. So we're trying, and really again what we're trying is to do everything that you shouldn't do. We have a site where we moved from a whole bunch of soil from an infested field soil site, spread it out, and now we're just going to plant peas there every single year until we actually see that inoculum has kind of proliferated enough to get those that site. So what's interesting though is that it seems like it's going to take a lot of soil to actually get it to establish. So we're always talking about sanitation and making sure you don't move soil. And I guess then if we're like, well, how much soil do you actually have to move to get Aphanomyces to establish? So that's kind of an interesting question. And we're hoping to also use that nursery to then see, okay, well, how have these fields become infested? If we started out with, you know, gallons and gallons of infested soil, how long does it actually take until it's become established in this area? So it's kind of, we're using it as a little research tool as well to see how the disease establishes. But I do have some plants from the greenhouse to show here. And this is really what Phanomyces root rot looks like. And you can, we didn't have great symptoms of Fusarium root rot, but when you look at something like this, oh, don't want to start spreading a Phanomyces root rot around here. These are kind of the really classical symptoms, and again, I'll pass this around. So honey, really honey brown discoloration of the roots. It moves all the way up to the epicaudal, and then it stops right at that green line, or sorry, right at the green stem. So kind of right at soil level is where we see the infection stop. And compare that to Fusarium, where you see infection start as kind of little points along the taproot, and then it becomes black later on. Phanomyces tends to stay kind of this honey brown color. The biggest problem is that Fusarium also seems to love roots that are infected by Phanomyces, so it'll go on and infect on top of the Phanomyces, and then you get these roots that are kind of brown and black, and it's really hard to figure out what's going on. But kind of in a perfect world, this is what a Phanomyces looks like, and I can pass those around. I got quite a bunch of these ones. And I should say that if anyone follows me on Twitter, they will see that I just posted, I think, yesterday that we are starting to see Phanomyces pop up in some fields. These are mostly at our field trial sites, so they're sites that we've chosen because they have high disease pressure. But now is actually a really good time if you're worried about your peas, or you have a history of growing peas and worried you might have a Phanomyces. Now is actually a really good time to go and dig up some of those roots, because early in the season is when we can really distinguish a Phanomyces from Fusarium. Right now, when you dig up a plant from a field, it's looking more like those classical symptoms, whereas if you wait until later in June, early July, when you start to see some evidence of root rot on your shoots and you dig up those plants, you usually can't tell if it's a Phanomyces or Fusarium at that point. So it's really kind of early season scouting where we can distinguish these two different pathogens. And I think these are all healthy roots. People probably don't want to see that. The other thing that we have here is just showing kind of the early, what a Phanomyces looks like when it just starts to infect. And these are like, these greenhouse plants are grown in vermiculite, so they're perfectly clean and white. We never see them like this coming out of the field. But these are a good example of just showing how Phanomyces starts, where you usually just start to see little bits of browning kind of on the lateral roots. It eventually kind of chews up all those lateral roots and then moves into that main stem and the epicaudal, and that's where we really see the damage start to occur. So I can pass these around if anyone wants to see kind of early, very low level symptoms of a Phanomyces. Thank you, yeah. Shouldn't have told you.
If you were digging your own plants in your field,
yeah,
like...
yeah, and we often, I should say, we only see that really early symptoms in the greenhouse. In the field, it's really hard to see that low level infection level, because you pull up the roots and they're kind of stained already from the soil, and it's hard to tell if this is an early season, just an early start of infection, or whether it's just dirty roots, really. So greenhouse, classical symptoms.
Okay,
yeah.
What can I do at this stage?
Nothing.
Okay.
Yeah. So with our root rots for both Fusarium and Phanomyces, we tell you to scout for them, and really it's scouting for them for the next time you're going to make a decision to plant a pea or a lentil crop. Because when you find the disease in your crop, there's nothing that you can spray for. There's nothing you can spray on your crop to help manage the disease below ground. There's no products, no fungicide products that move from the shoot down into the root. So it's really just knowing what's in your field, and then knowing how you have to maybe manage that field the next time you're going to choose to plant pea or lentils. So this really, the management of this disease is really key is based on avoidance of infested fields. And then the other issue, it would be planting partially resistant cultivars when we have them, and we don't have those yet. Lots of breeding programs are actively working on trying to incorporate Aphanomyces and Fusarium Root Rot resistance into some of our agronomically acceptable lines. So,
would the combine monitor tell me already?
Well, you would probably be able to see areas of depressed yield in your field, and really what you want to watch for is areas that are maybe starting to show some yellowing patches in the field, and when you're combining you notice, okay, this area has much lower yield, and then you might want to go and you can do some soil testing of that site to see if maybe it's because you have Aphanomyces or Fusarium Root Rot in that area. And you can send tissue samples and soil samples, I think, to both BioVision and 2020 Seed Labs, and they do testing for Aphanomyces. I don't think they do testing for Fusarium yet, because that one is actually, it turns out it's a little bit harder to test for with a molecular test. But yeah, so it's really, prevention is kind of the key to management here. Yeah, or avoidance, I should say. Yeah, I know, it's a tough one. It's a tough one to work with, and that's why really breeding for resistance is going to be the best tool that we have. It's just maybe we'll take some time to get that resistance into our lines. Yeah.
So same question as before, other hosts?
Aphanomyces is specific to legume crops, and within the legumes it's only seems to be specific to very specific pulse crops. So it likes peas and lentils. Soybean is not a host. Faba beans don't appear to be a host. Chickpeas also don't appear to be a host. And we have a research project going on right now looking at including some of these non-host pulse crops in rotations to see what the effect will be. So we want to make sure that if we're recommending I'll plant a soybean instead of a pea, that the soybean aren't going to help kind of continue the inoculum levels in the field, that they will in fact act as a non-host and we won't see any infection at all. So we have some rotation trials going on with those three pulse crops, chickpeas, soybean, and faba beans right now, just to confirm that. We've done greenhouse testing, but it's always good to bring things out into the field. Yeah.
How about dry beans?
Dry beans are also not a host. All pathogens have a very complicated kind of rating system, and I know when Reem talks about rust, she'll talk a lot about races. So there are different races or pathotypes of Aphanomyces, and there is a race that can infect beans, but we don't seem to have it here. And usually the pea races cannot infect dry beans, but a dry bean race might be able to infect peas, but we don't seem to have it in dry beans. So for now, we're good there. Yeah. Oh, good. Christine is giving a nice demo over there. Yeah. And I'm not sure how I'm doing for time, but the last thing I wanted to just talk about and jump over was talk about the other screening that we do in my pulse crop pathology program, and that's we do a lot of screening for dry beans, white mold on dry beans. So we have a disease nursery as well here on station. It's kind of a couple rows down over there, where we are screening dry bean lines for white mold. And I'm not sure how many of you here are dry bean growers. Probably not a lot. A few, yeah. And so if you grow dry beans, then you know that your biggest issue is really dealing with white mold. But if you grow dry beans, you probably also are growing canola, or if you don't grow dry beans, you're probably growing canola. And it's the same pathogen that causes white mold in canola, that causes sclerotinia, stem rot. Sorry, white mold in dry beans, the same pathogen that causes sclerotinia, stem rot in canola. Yeah. They just have given them two different disease names, but it's the same pathogen. So you should be, so I think this project kind of might also have some benefits to canola. But what we do is we screen dry beans for white mold in the field, and that's to see if they have field avoidance to white mold. So is there aspects of their agronomy that's helping them avoid the disease? But what most of our dry bean lines do not have is any genetic resistance. So to screen for genetic resistance, we have to do a greenhouse screening. And I've brought some plants out here to show how we screen for white mold for genetic resistance. And these are very commonly grown lines here in Alberta. I think one of them is Black Diamond, and one of them might be Island or Resolute. And we can see that there is absolutely no genetic resistance in these lines. Like, the plants are completely destroyed. And then in the back here, we have some germplasm lines that do have genetic resistance to white mold. And so what we do is we infect one little stem with some of the white mold pathogen, and then see how far down it's moving down that stem. So for these ones, we can see it's moved completely down the stem and the plants are dead. Whereas for our germplasm resistant lines, we can see that it kind of stops at that first node and it can't move down. So now a major kind of press from the dry bean breeding program is to try and incorporate this resistance from germplasm lines into lines that can be grown in southern Alberta. The problem is that a lot of these lines with genetic resistance have very poor growth habit. A lot of them are really viney. They are maybe photoperiod insensitive, so you can't grow them in our northern climates. And so it's a real challenge to try and move that genetic resistance into some of these lines. But our bean breeder, Partiva, is really actively working on this. And so we screen all the lines that come out of his breeding program to see if any of them are showing any genetic resistance as well as field resistance. And that will hopefully really help with some of the white mold problems that we have down here. And then the last thing that we're doing with regards to white mold and really Sclerotinia is those little spore samplers that you see behind you. Yeah. So if you come really close, you can kind of hear them. But what it is, take the lid off. Oh, it doesn't come off very easily. There we go, yeah. So what it is this thing just kind of makes sure that it's kept into the wind. But then we have an active little motor here that is actively sucking in air through this little air intake. Everything that's coming in, that's getting sucked in through the air, gets deposited into one of these little vials. And then this is set to move over every day. So every day we can collect a new air sample into these vials. And then we take the vials back to the lab and do a DNA analysis to be able to quantify how much Sclerotinia is flying around in the air. And what we're trying to work on now is whether we can get this to act as kind of a real-time process. So can we get kind of these high throughput techniques going so that we can kind of look at samples from these samplers that are spread all across southern Alberta? Can we get data out within kind of 24 to 48 hours of a sample being collected? And then use that as kind of a warning system or a forecasting system for producers to say, okay, there's lots of Sclerotinia ascospores flying around in the air right now. If your dry beans are flowering or if your canola is maybe at the susceptible stage, you should consider putting on a fungicide. And then alternatively, if a lot of times growers will just go and put a fungicide in when their dry beans are flowering, start to flower, alternatively we can say, hey, there's actually no Sclerotinia flying in the air right now, so your risk might be fairly low. Maybe you don't need to apply your fungicide at this time. Yes, yeah, and what we're hoping to do is, like, because I work on pulses, I'm focusing on Sclerotinia and pulses, but what we're hoping to do is maybe try to expand this to look at, can we assess for multiple pathogens at a time? Because really these samples are collecting everything that's in the air, right? So we could start screening for rust, for Fusarium graminearum, for Sclerotinia. Yes, yeah. Yeah, and because there's some passive samplers that just rely on the wind kind of moving them through, but this is sucking in the air, and it sucks in a known volume of air too at a time, so you can then go back and say, you have this many spores per cubic meter of air. Yeah, yeah. The only problem with this system, it's a company in the UK that makes these, and it's all kind of 3D printed plastic, which seems to be a little bit sensitive sometimes, so they can sometimes easily break, and then we have to send them all the way back to the UK to get repaired. That's really been our only challenge with these, is they're not always working when you want them to work. But we hope to just have enough running that there's always kind of a backup there. And this is really just, we kind of ran this as a pilot project last year, and then this year is really when we're getting into the research. So we want to see how well we are actually being able to track these ascospores and how well that's correlating to the disease pressure that we're seeing in the fields. And then hopefully see if we can expand it to maybe an Alberta-wide network.
Will it only 24 hours? Will it run all night?
Yes, yeah. And you know what we could do, you can set the timer however you want. So if we wanted to, we could collect samples every 2 hours and then change it over to a new vial. Because a lot of these samplers are, out in Bow Island and far away from us, we're just looking at daily samples. But yeah, you can set it for basically 24 hours. Yeah.
So are you collecting data on them now? Is there anything showing up?
No, so we just put these out yesterday because it's still a little bit early for disease, and then we're just testing to make sure that they're working, and then we'll start pretty soon. Yeah, and then one of the challenges too, I guess, is figuring out then how to communicate this information to producers. What sort of technology do we want to use? And that's something that we'll hopefully incorporate into the project, besides me just tweeting out results, and it's probably not the most efficient way to do it, but yeah.
Are you taking those samples for everybody? Like, is it going to be Fusarium people can look at it? No.
Well, yes, like for right now we are focusing on Sclerotinia, but we do have a proposal in, I guess we don't know whether it'll get funded or not, to expand it to a multi-pathogen, multi-crop forecasting network.
Now you're storing samples, right?
Yes, yeah. And we're working with a lot of the other pathologists. So if I only work with Sclerotinia, we're working with, like Mike Harding is doing a very similar project with late blighted potato, and I think Kelly Turkington is doing a project on Sclerotinia and canola. I don't know if anyone's actually working on Fusarium of wheat, but it's definitely something that we can.
Not too many rainy years.
Yeah.
So everybody will complain.
Yeah, exactly. And even last year when we had this out, it was such a dry year. We really didn't see a lot of ascospores or white mold either. So it's really dependent on the weather. And that is the other thing that we're hoping to do. Everywhere we have a spore sampler, we also have a weather station. And so we are looking to see, well, can we start using weather to predict when the spores are being released, so we don't always have to rely on actually monitoring spores, if we can come up with some weather models. And they have some good weather models in the States for white mold on soybean. So it's something too that we'll look at, okay, can we use that same model and apply it to white mold of dry bean, and then expand that to canola and things like that. And I know for Fusarium there is kind of a weather-based forecasting model as well, right? But not a spore-based model at this time. Yeah. So any questions, I guess, on?
How accurate would the sample it take be for, let's say, 100 kilometers away? Would it be, do you guys know yet how big of a distance that's accurate for?
That's one of the big questions that we're looking at. And so this year what we're actually doing is putting a whole bunch of samplers in a couple of bean fields and saying, okay, well, is there a big variation between one end of the bean field versus the other end? And then we have samplers outside of the bean field and saying, okay, how is that relating? So that is one of the big questions, is what's the kind of the regional spread or density of these samplers that you need to be accurate? Yeah.
Wouldn't the best work want the multiplication of the wind? Like 5 mile an hour wind we had yesterday with so much square.
Yeah, and I guess we talked to some of the weather modelers more about that. And really what we're interested in is are the conditions right kind of at the soil surface that we're actually getting spore release? And then once you get that spore release, then the next question would be, well, then how far are they actually moving? And for Sclerotinia, most of the spores are a little more localized, where they're kind of, it's spores that are coming from your own field that are likely to infect your field. Whereas I think with something with Graminearum, they might move a little more on the wind, and same with, I think, Rima's around here somewhere. Like with rust spores, they're more likely to be moving in the wind, whereas Sclerotinia is pretty kind of localized. So that's something that we're thinking about as well. Okay. Yes, thank you.